Good evening, guys. Just let me know once if you're able to see my screen. There's some kind of network issue from my side. Everything is lagging behind. Can you see my screen? Anyone? What's the problem? There's some kind of network issue. Let me check myself. Okay, you can see my screen. I was checking myself. Yeah. So I don't know. There's some kind of network issue from my side. So I might be disconnected today because I don't know what's happening. Everything is lagging behind the entire day, like some kind of network failure in the campus. So in case something like that happens, I'll still continue speaking. I'll upload the video later, so you can check the video on YouTube if you want. Yeah, definitely some kind of network issue. Anyway, but you can see these slides, right? Okay, so he has replied yes. So I believe from this slide is visible. Okay, so we'll start today's session. So if at any point I become inaudible or if my slide doesn't show up, please let me know because everything is lagging behind. I don't know what's the issue. That's why I'm pretty sure that today there's gonna be some. There must have been some network issue. Yeah. Okay. So good evening, everyone. Someone else. Yeah. So good evening. This is the 12th session of this live session of this course, Applied Environmental Microbiology. My name is Aishi and I am a second year PhD student in the Department of Chemistry at IIT Bombay, and I am your NPTEL TA for this course. So as like we did in the previous sessions, today also we'll be discussing some of the sample problems this year's or last year's, and we'll be discussing some of the answers, and hopefully that will help you solve questions in the main exam, which will be happening in a week, I guess. I'm not really sure about the date. So let's start with the first question for today. Okay, so disease A has 82% mortality rate in a densely populated area and is transmitted by aerosol. Disease B has 89% morbidity rate and is spreading through oral fecal route by a pathogen which can survive in unfavorable conditions for extended duration. Given this, which of the following can be said about the R naught value? Option A, R naught of disease B will be lower than disease A. R naught of disease A will be 0. R naught of disease B will be 1, or none of the above. What do you think is the right answer? You can try answering. So the first thing that should come to your mind is how is R naught related to the morbidity or the mortality rate. If you can figure that out, then maybe this answer is okay. Sorry, you can already see my answer. I did not check before. Wait. Okay. So I forgot to put the animation. Sorry. Anyway, so now you have already seen the answer. Yeah. So it's actually none of the above. Does anybody want to? Does anybody have any different opinion, or do you agree with none of the above? So basically R naught, which is R0 or R naught here, is a mathematical term that indicates how contagious or infectious a disease is, basically. Okay, so it's also referred to as a reproduction number. So as an infection is transmitted to new people, it reproduces itself. So R naught basically tells you the average number of people who will contract a contagious disease from one person with that disease. It specifically applies to a population of people who were previously free of infection and haven't been vaccinated. For example, if a disease has an R naught value of 18, a person who has a disease will transmit it to an average of 18 other people. Okay, and that replication will continue if no one has been vaccinated against the disease or is already immune to it in their community. So, three possibilities basically exist for the potential transmission or decline of a disease, depending on the R naught value. So, if R naught is less than 1, each existing infection causes less than one new infection. In this case, the disease will eventually decline and eventually die out. If R naught equals God, there's some. Did I accept? There's some kind of network issue from my side. Anyway, I hope my screen is visible. If my screen becomes if my screen at any point becomes doesn't show up, please let me know once via message or just unmuting yourself. Anyway, I'll be recording this. I'm recording this session and will be uploading it, so it doesn't matter. So yes, so as I was saying, the R naught value basically tells you the average number of So the R naught value basically tells you the average number of people who will contract a contagious disease from one person with that disease. So it specifically applies to a population of people who were previously free of infection and haven't been vaccinated yet. For example, if a disease has an R naught value of 18, a person who has a disease will transmit it to an average of 18 other people, and that replication will continue if no one has been vaccinated against the disease or is already immune to it in the community. Okay, so what do these R naught values mean? So three possibilities exist for the potential transmission or decline of a disease depending on its R naught value. So if R naught is less than 1, each existing infection causes less than one new infection, and in this case the disease will decline and eventually die out. If R naught equals 1, each existing infection causes one new infection. The disease will stay alive and stable, but there won't be an outbreak or an epidemic. But if R naught is more than 1, each existing infection causes more than one new infection, and the disease will be transmitted between people and there may be an outbreak or an epidemic. So, importantly, a disease's R naught value only applies when everyone in a population is completely vulnerable to the disease. This means that A, no one has been vaccinated, B, no one has had that disease before, and C, there's no way to control the spread of the disease. Okay? So this combination of conditions is rare nowadays, thanks to the advancements in medical science, and many diseases that were deadly in the past can now be contained and sometimes even cured. For example, in the 1918, there was a worldwide outbreak of the swine flu that killed approximately 50 million people. So, according to a review article that was published in BMC Medicine Trusted Source, the R naught value of the 1918 pandemic was estimated to be between 1.4 to 2.8. Okay, but when the swine flu or the H1N1 virus came back in 2009, its R naught value was between 1.4 and 1.6. Report researchers in the journal Science Trusted Source of thing. So the existence of vaccines and antiviral drugs made the 2009 outbreak much less deadly. So that's why the R naught value has comparatively decreased. So the in case of COVID-19, if you think about COVID-19, which is like the pandemic that happened basically. So the R naught value for COVID-19 was a median of 5.7, according to a study published online in the Emerging Infectious Diseases. So that's about double an earlier R naught estimate of 2.2 to 2.7. So this 5.7 R naught value means that one person with COVID-19 can potentially transmit the coronavirus to 5 to 6 people. Okay, rather than 2 to 3, as researchers originally thought. And researchers calculated the new number based on data from the original outbreak from Wuhan, China. So they used parameters like the virus incubation period, 4.2 days, how much time elapsed from when people were exposed to the virus and when they started to show the symptoms. So the researchers estimated a doubling time of 2 to 3 days, which is much faster than the earlier estimates of 6 to 7 days. So the doubling time is how long it takes for the number of coronavirus cases, hospitalizations, and deaths to double. And the shorter the time, the faster the disease is spreading. So with an R naught value of 5.7, at least 82% of the population needs to be immune to COVID-19 to stop its transmission through vaccination and herd immunity. So the study authors say active surveillance tracking the contacts of people who contracted the coronavirus, quarantining people, and strong physical distancing measures were needed to stop the coronavirus from being transmitted. So how is this R naught value of a disease calculated? So there are a few factors that are taken into account to calculate the R naught of a disease. The infectious period. So some diseases are infectious or contagious for a longer period. Hi, my voice is breaking. I am really sorry. I don't know why. There must be some kind of network issue. Wait, let me write. I don't know. I'll write it down. The laptop is lagging a lot since the morning. So if you can hear me now, there's some kind of network issue from my side only. I understand that. Uh, I'm not able to like sort it out. But anyway, I'm recording the session from a third party software. So anyway, it will be recording the entire session and I'll upload it to YouTube. So in case you want to refer to it later, you can obviously find the. I'm sorry, my voice is breaking. There's some. See, I'm done typing, but it's still not showing up. Adults with the flu are typically contagious for up to 8 days, and children can be contagious for longer than that. So the longer the infectious period of a disease, the more likely a person who has it can transmit the disease to other people. And a long period of infectiousness will contribute to a higher R naught value. Another factor that it depends on is the contact rate. So, if a person who has a contagious disease comes into contact with many people who aren't infected or vaccinated, the disease will be transmitted more quickly. And if the person remains at home in the hospital or otherwise quarantined while they are contagious, the disease will transmitted much more slowly, and a higher contact rate will contribute to a higher R naught value. I'm sorry, there's some kind of network issue because of which I'm getting thrown out of the meeting. Okay, I'll share my screen again. Hopefully this time there will be lesser network issue. Please let me know if my screen is visible. Can you guys hear me? Is my screen visible?
No, ma'am. Voice are breaking, ma'am.
Okay, screen is also not visible.
Yes, ma'am.
Okay. So there's some kind of internet issue from my side due to which I think. Is my screen visible now?
Yes, ma'am.
Okay, okay. So yeah. So we were talking about R naught value. So we were talking about the factors on which the R naught value depends. Like how is the R naught value of a disease calculated? So the first factor that determines the R naught value is the infectious period. So some diseases are more contagious. Some diseases are contagious for longer periods than others. For example, according to the Centers for Diseases Control and Prevention trusted source, adults with the flu are typically contagious for up to 8 days. Children can be contagious for longer than that. So, the longer the infectious period of a disease, the more likely a person who has it can transmit the disease to other people. A long period of infectiousness will contribute to a higher R naught value. Another factor can be contact rate. Okay, so if a person who has the contagious disease comes into contact with many people who aren't infected or vaccinated, the disease will be transmitted more quickly. But if that person remains at home, in a hospital, or otherwise quarantined while they are contagious, the disease will be transmitted much more slowly. A high contact rate will contribute to a higher R naught value. So that is why during the COVID-19 pandemic, we were asked to stay at homes and not like physically distance ourselves basically. Another factor that determines the R naught value is the mode of transmission. The diseases that are transmitted the fastest and the easiest are the ones that can travel through air, such as flu or measles. So, physical contact with the person who has such a disease is not needed to transmit it. You can contract the flu from breathing near someone who has the flu, even if you never touch them. But in contrast, diseases that are transmitted through bodily fluids such as Ebola or HIV, those really aren't as easy to contract or transmit. This is because you need to come into contact with infected blood, saliva, or any other bodily fluids to contract them. So airborne illnesses tend to have a higher R naught value than those spread through direct contact. So what conditions are basically measured by this R naught value? R naught can be used to measure any contagious disease that may spread in a susceptible population. Some of the most highly contagious conditions are measles and the common flu. And more serious conditions such as Ebola and HIV spread less easily between people. So this illustration basically shows some commonly known diseases and their estimated R naught value. So you can see how measles has an R naught value of 18. It can spread so easily. mumps has an R0 value of 10, SARS has an R0 value of 4, and so on and so forth. So what are the tips for prevention? So R0 is actually a very useful calculation for predicting and controlling the transmission of disease, and medical science continues to advance. So researchers are discovering new cures for different conditions, but contagious diseases aren't going to disappear anytime soon. So what steps can you take to help prevent the transmission of contagious diseases? So you have to, at the first, education is the most important. You have to learn how different contagious diseases are transmitted. Okay, so you have to know if it's transmitted through bodily fluids, or if it's an airborne transmission. You have to know that. Or you can ask any medical professional, any scientist or doctor about steps you can take to stop the transmission of the infection. For example, during the COVID-19 pandemic, we were regularly asked to wash our hands routinely with soap and water, especially before we eat or prepare food, or actually before we touch our hands to our face or eyes, basically any point where our hands are getting in contact with inner layers. Also, we have to stay up to date with routine vaccination, and we should ask our doctor what diseases we should be vaccinated against. Okay, so but how is R0 value related to the mortality rate? So this question was about basically mortality rate, right? So R0, as I said, is a term used in epidemiology to represent the basic reproduction number of a virus or disease. It refers to the average number of people who will contract the disease from one infected person in a population where everyone is susceptible to the disease and no interventions such as vaccinations or social distancing are being implemented. So the disease mortality percentage or case fatality rate. This is a mortality percentage. It is also called case fatality rate. One second. So this mortality percentage, also known as case fatality rate, is basically the proportion of people who die from a specific disease among all individuals diagnosed with the disease. So this mortality rate can vary depending on factors such as age, underlying health conditions, and the availability of medical treatment. So the R0 and the mortality rate values are related in that the R0 can influence the overall impact of a disease outbreak. If the R0 value is high, the disease can spread rapidly and affect a larger number of people, which may increase the absolute number of deaths even if the mortality rate is relatively low. Conversely, if the R0 value is low, the disease may spread more slowly and the absolute number of deaths may be lower even if the mortality rate is relatively high. So it's important to note that the R0 and the mortality rate are not the only factors that determine the impact of a disease outbreak. Other factors such as the effectiveness of medical treatments and public health interventions are also equally important. So, is it possible to determine the R0 value if we know the case fatality rate or the mortality rate? No, it's not possible to determine the exact R0 value of a disease outbreak from case fatality rate or mortality rate alone. Because case fatality rate represents the proportion of people who die from a specific disease among all individuals diagnosed with the disease. In contrast, R0 is a measure of how contagious a disease is, representing the average number of people who will contract the disease from one infected person in a population where everyone is susceptible to the disease and no interventions such as vaccinations or social distancing are being implemented. So the R0 value can be estimated using mathematical models that take into account factors such as the infectiousness of the disease, the length of the infectious period, and the mixing patterns of the population. So the case fatality rate alone does not provide enough information to estimate the R0 value, as it only reflects the severity of the disease in terms of its impact on those who are diagnosed with it. Therefore, while both R0 and case fatality rate are important measures of the impact of a disease outbreak, they represent different aspects of the disease and cannot be used interchangeably or to estimate one another. So that is why you cannot estimate or assume any of these options based on the case fatality rate, because both of these are important but they represent different aspects of the disease, so they cannot be used interchangeably or they cannot be used to estimate one another. So that is why none of the above options are correct. Okay, I am still in the meeting, right? Yeah. Okay, I hope my voice is clear and screen is also visible. Let me know if my screen stops showing at any point. So we'll move on to the next question. The human body communicates with the outside world through natural biosensors. Is it true or false? Anybody? The statement the human body communicates with the outside world through natural biosensors. Is it true or false? So, this statement is true to a certain extent. So, the human body has several natural mechanisms that sense and respond to various stimuli from external environment, right? Including touch, smell, taste, hearing, and vision. So, these mechanisms involve specialized sensory receptors located throughout the body that detect changes in the environment and transmit signals to the brain, which interprets the information and generates appropriate responses. For example, the skin contains specialized sensory receptors called mechanoreceptors, which respond to touch and pressure, as well as thermoreceptors, which respond to changes in temperature. So, the nose contains olfactory receptors that detect various smells, while the tongue contains taste buds that detect different tastes such as sweet, sour, bitter, and salty. And the ears contain specialized receptors that detect sound waves, while the eyes contain photoreceptors that detect light and allow us to see. So, in addition to these specialized sensory receptors, the human body also has natural biosensors that detect and respond to changes in various physiological parameters such as heart rate, blood pressure, and oxygen levels. For example, the baroreceptors located in the walls of the heart and the blood vessels sense changes in blood pressure and send signals to the brain, which then adjust the heart rate and blood vessel diameter to maintain stable blood pressure. So the human body also has natural biosensors that detect changes in the glucose levels, hormone levels, and other biochemical parameters. For example, the pancreas contains specialized cells called beta cells that secrete insulin in response to changes in the glucose levels in the blood. So overall, the human body has various natural biosensors that sense and respond to changes in the external environment and internal physiological parameters. These mechanisms allow the body to maintain homeostasis, adapt to changes, and respond to various stressors and challenges in the environment. Okay, so this statement is true to a certain extent, definitely. Moving on to the next question. Removal of which part of biosensor will make it impossible to interpret the result but will still be able to receive the signal? Which one do you think is correct? Anyone? Anyone? I'm in the meeting, right? Okay. So, the answer will be transducer. So, before that, we'll talk a little bit about biosensor. We already did talk about it a lot last week, but let's recapitulate. So, biosensors are nowadays really ubiquitous in biomedical diagnosis as well as a wide range of other areas such as point of care monitoring of treatment and disease progression, environmental monitoring, food control, drug discovery, forensics, and biomedical research. So, a wide range of techniques can be used for the development of biosensors. Their coupling with high affinity biomolecules allows a sensitive and selective detection of a range of analytes. So I'll give you a general introduction to biosensors and biosensing technologies, including a brief overview, introducing key developments in the field and illustrating the breadth of the biomolecular sensing strategies and the expansion of nanotechnological approaches that are now available. So, a biosensor is basically a device that measures biological or chemical reactions by generating signals proportional to the concentration of an analyte in the reaction. So, biosensors are employed in applications such as disease monitoring, drug discovery, and detection of pollutants, disease causing microorganisms, and markers that are indicators of a disease in bodily fluids. Okay, so this is a typical biosensor representation. It consists of the following components: analyte, which is a substance of interest that needs detection. For example, glucose is an analyte in a biosensor designed to detect glucose. Then we have bioreceptor. A molecule that specifically recognizes the analyte is known as bioreceptor. So enzymes, cells, aptamers, deoxyribonucleic acid, and antibodies are some examples of bioreceptors. And the process of signal generation in the form of light, heat, pH change, charge or mass change can, upon interaction of the bioreceptor with the analyte, is termed as biorecognition. Okay, then we have the transducer. So the transducer is an element that converts one form of energy into another. So in a biosensor, the role of the transducer is to basically convert the biorecognition event into a measurable signal, and this process of energy conversion is known as signalization. And most transducers produce either optical or electrical signals that are usually proportional to the amount of the analyte bioreceptor interactions. Then we come to the electronics parts. So this is the part of a biosensor that processes the transduced signal and prepares it for display. It consists of complex electronic circuitry that performs signal conditioning such as amplification and conversion of signals from analog into digital form, and the process signals are then quantified by the display unit of the biosensor. So the display consists of a liquid, that's our user interpretation system, which is a liquid crystal LCD, basically a liquid crystal display of a computer or a direct printer that generates numbers or curves understandable by the user. And this part often consists of a combination of hardware and software that generates results of the biosensor in a more user-friendly manner. So the output signal on the display can be numeric, graphic, tabular, or an image depending on the requirements of the end user. Okay, so there are several characteristics of a biosensor as well. Like there are certain static and dynamic attributes that every biosensor possesses, and the optimization of these properties is reflected on the performance of a biosensor. Such a criteria is selectivity. So the selectivity is perhaps the most important feature of a biosensor, because selectivity is the ability of a bioreceptor to detect a specific analyte in a sample containing other admixtures and contaminants. So the best example of selectivity is depicted by the interpretation of an antigen with the antibody. Classically, antibodies act as bioreceptors and are immobilized on the surface of the transducer. A solution, which is generally a buffer containing salts containing the antigen, is then exposed to the transducer where the antibodies interact only with the antigens. And to construct a biosensor, selectivity is the main consideration actually when choosing biosensors. Okay, so this is one condition or one criteria for selectivity for biosensors. Another criteria is reproducibility. So reproducibility is the ability of the biosensor to generate identical responses for a duplicated experimental setup. The reproducibility is characterized by the precision and the accuracy of the transducer and electronics in the biosensor. And precision basically is the ability of the sensor to provide alike results every time a sample is measured, and the accuracy indicates the sensor's capacity to provide a mean value close to the true value when a sample is measured more than once. Okay, so reproducible signals provide high reliability and robustness to the interface made on the nature of the biosensor. Another criteria is obviously stability. The stability is the degree of susceptibility to ambient disturbances in and around the biosensor system. So these disturbances can cause a drift in the output signals of a biosensor under measurement, and this can cause an error in the measured concentration and can affect the precision and the accuracy of the biosensor. So stability is the most crucial feature in applications where a biosensor requires long incubation steps or continuous monitoring. So the response of transducers and electronics can be temperature sensitive, okay, because which may influence the stability of the biosensor. So therefore appropriate tuning of electronics is required to ensure a stable response of the sensor. Another factor that can influence the stability is the affinity of the bioreceptor, which is the degree to which the analyte binds to the bioreceptor. Bioreceptors with high affinities encourage either strong electrostatic or covalent linkage of the analyte that fortifies the stability of the biosensor. Another factor that affects the stability of the measurement is the degradation of the bioreceptor over a period of time. Then another criteria is sensitivity, obviously. The minimum amount of analyte that can be detected by a biosensor defines its LOD, or limit of detection sensitivity. In a number of medical and environmental monitoring applications, a biosensor is required to detect nanogram per ml or even femtogram per ml to confirm the presence of traces of analytes in a sample. For example, a prostate specific antigen called PSA concentration of 4 mg per ml in blood is associated with prostate cancer, for which doctors suggest biopsy tests. Hence, sensitivity is considered to be an important property of biosensor. Another important property is linearity. So, what do we exactly mean by linearity? It's basically the attribute that shows the accuracy of the measured response. For a set of measurements with different concentrations of analyte to a straight line, mathematically represented as y equal to mc, where c is the concentration of the analyte and y is the output signal, and m is the sensitivity of the biosensor. So, the linearity of the biosensor can be associated with the resolution of the linearity can be associated with the resolution of the biosensor and range of analyte concentrations under test. So, the resolution of the biosensor is defined as the smallest change in the concentration of an analyte that is required to bring a change in response of the biosensor. So, depending upon the application, a good resolution is required, as most biosensors, applications require not only analyze detection but also measurement of concentrations of analyte over a wide working range. So another term associated with which is termed as defined as the linearity. Another term associated with the linearity in the linear range, which is defined as the range of analyte concentrations for which the biosensor response changes linearly with the concentration. So, biosensors have actually had a wide range of applications that aim to improve the quality of life. So, this range covers their use for environmental monitoring, disease detection, food safety, defense, drug discovery, and many more. And one of the main applications of biosensor is basically the detection of molecules that are either indicators or for disease or targets of a drug. For example, electrochemical biosensing technique can be used as a clinical tool to detect protein cancer biomarkers. Okay, and biosensors can also be used as platforms for monitoring food traceability, quality, safety, and nutritional value. So these applications fall into the category of single shot analysis tools, that is where cost effectiveness and disposable sensing platforms are required for the application. On the other hand, an application such as the pollution monitoring requires a biosensor to function from a few days to few hours to several days, and such biosensors can be termed as long-term monitoring and analysis tools. Whether it is long-term monitoring or single-shot analysis, biosensors find their use as technologically advanced devices both in the resource-limited setting and sophisticated medical setups. Example with applications in drug discovery and for the detection of a number of chemical and biological agents that are considered to be toxic materials, materials of defense interest, for using artificial implantable devices such as pacemakers and other prosthetic devices. So a range of electrochemical, optical and acoustic sensing techniques have been utilized along with the integration into analytical devices for various applications. So the question was, removal of which part of biosensor will make it impossible to interpret the result and but still be able to see the signal. So in a biosensor, the signal is generated by a biological recognition element, okay, which specifically interacts with the target analyte and generates a measurable signal. The signal is then transduced by a physical or a chemical sensor elements into a measurable output signal, which is then proceeded and interpreted by an electronic or digital component. So if the recognition element of a biosensor is removed, it would not be able to specifically detect the target analyte, and therefore the biosensor would not be able to generate a meaningful signal. Thus, removing the recognition element would make it impossible to interpret the results and receive the signal. On the other hand, if the physical or chemical sensor element is removed, the biosensor may still be able to generate a signal in response to the interaction between the recognition element and the target analyte. However, the signal would not be transduced into a measurable output signal, and therefore it would not be possible to interpret the result even though the biosensor is still able to receive the signal. So therefore, it is the removal of the physical or the chemical sensor element of a biosensor that would make it impossible to interpret the result while still being able to receive the signal. Okay, so yes, so when I mentioned that there's something called the physical or chemical sensor element, I was actually referring to the transducer. Okay, component of the biosensor. Transducer converts the energy measurable output signals such as an electrical or optic signal. So if the transducer is moved away from the biosensor, the recognition element will still be in contact with, in contact the target analyte and generate a signal, but it would not be converted into a measurable output signal that can be interpreted. Therefore, removing the transducer would make it impossible to interpret the results while still being able to receive the signal. So this is what the question was all about. So the answer is transducer. Okay. Okay, next question. With respect to SIRS, which of the following is false? SIRS is surface enhanced uh, Raman spectroscopy. Yeah. So which one do you think is false with respect to SIRS? Anyone? I already showed the answer, but anyone? Last option. Yeah. Why do you think it's none of the above? All of these are true for surface enhanced Raman spectroscopy. So you're saying that none of the above is false. All of these are true. Okay. Yeah. So basically, surface enhanced Raman scattering, basically, not spectroscopy. We can call it scattering. So surface enhanced Raman scattering, sorry, is called SIRS, and it's a very powerful analytical technique that enhances the Raman scattering signal of the target molecules by using nanostructured, metal surfaces such as silver or gold, basically, to amplify the Raman signal. So the Raman effect is a scattering process that occurs when a photon of light interacts with the molecule, causing the molecule to vibrate and produce a scattered photon with a different energy and wavelength than the incident photon. So the Raman spectrum of a molecule provides a unique spectral fingerprint that can be used for the identification or the and the quantification of the molecule. So in surface enhanced Raman scattering, the target molecules are adsorbed onto the metal surface and the incident laser light interacts with the metal nanoparticles, causing the formation of localized surface plasmons. The electric field of the plasmons interacts with the target molecules, enhancing their Raman scattering signal by several orders of magnitude. So the enhancement of the Raman signal in SERS, like in surface enhanced Raman scattering, depends on several factors, including the size, shape, and the composition of the metal nanoparticles, as well as the nature of the target molecules and the interactions with the nanoparticles. So the interaction between the target molecules and the metal nanoparticles can lead to a chemical enhancement, which are the where the metal surface catalyzes chemical reactions that enhance the Raman signal. Okay, so or electromagnetic enhancement, where the plasmon resonance of the metal nanoparticles enhances the electric field of the incident light, increasing the Raman scattering signal. And SIRS process can detect even trace amounts of target molecules with high sensitivity and specificity, making it a very powerful analytical tool for various fields of research and industry. Okay, including biomedical diagnostics, environmental monitoring, and food safety testing. SIRS-based biosensors have been developed in the detection of various molecules such as proteins, nucleic acids, and viruses with high sensitivity and specificity. Okay, so overall, the SIRS is a highly sensitive and specific analytical technique that contributes to the principle of Raman spectroscopy and nanotechnology, following the detection and quantification of specific molecules with high precision and accuracy. Okay. So the discovery of the enhancement of Raman scattering by molecules adsorbed on nanostructured metal surfaces is a landmark, I think, in the history of in the history of spectroscopic and analytical techniques. and significant experimental and theoretical effort has been directed towards understanding the surface enhanced Raman scattering effect and demonstrate its potential in various types of ultra-sensitive sensing applications in a wide variety of fields. So, in the 45 years since its discovery, SIRS has blossomed into a rich area of research and technology, but additional efforts are still needed before it can be completely routinely used analytically and in the commercial products basically. So there are a lot of reviews on online where you see prominent authors from around the world join together basically to summarize the state of the art in understanding and using SIRS and to protect what can be expected in the near future in terms of research applications and technological development. Okay, so this surface enhanced Raman scattering is very commonly used sensing technique in which the inelastic light scattering by molecules is greatly enhanced by factors of up to 10 to the power 8 or 10 to the power 9, even larger, enabling single molecule source in some cases. When the molecules are adsorbed onto the corrugated metal surfaces such as silver or gold nanoparticles. Since its discovery over 40 years ago, it has enjoyed steady growth of interest in the research community, and it has spawned a variety of other spectroscopic techniques basically that take advantage of enhanced local fields that arise from plasma excitation in the nanoparticles for optical phenomena such as fluorescence and nonlinear optics. And in addition, the coupling of SIRS with atomic force microscopy or scanning tunneling microscopy such as STM tips has led to the tip enhanced Raman scattering, which is a powerful, interesting tool. So, for analytical applications, SIRS can be differentiated from many other techniques by the rich vibrational spectroscopic information that it provides, which has led to applications in several different directions, including electrochemistry, catalysis, biology, medicine, art, conservation, materials science, and others. Okay, so basically it's nothing but a surface sensitive technique that enhances Raman scattering by molecules adsorbed on rough metal surfaces or by nanostructures such as the plasmonic magnetic cells nanotubes. The enhancement factor can be as much as 10 to the power 10 or 10 to the power 11, which means the technique may detect single molecules. Okay, so the exact mechanism of the enhancement effect of SIRS is still a matter of debate in the literature. There are two primary theories, and while their mechanisms differ substantially, distinguishing them experimentally has not been straightforward. The electromagnetic theory proposes the excitation of a localized surface plasmons, while the chemical theory proposes the formation of a charge transfer complex. And the chemical theory is based on the resonance Raman spectroscopy, in which the frequency coincidence of the incident photon energy and the electron transition greatly enhances in intensity. So research in 2015, I guess, on a more powerful extension of the SIRS technique called the slip SIRS or strategy liquid diffuse porous SIRS has featured engraved in the back of as further basically the research that was conducted in 2015 on a more powerful extension of the SIRS technique, which is called slip SIRS or slippery liquid infused porous SIRS, has further supported the electromagnetic theory. So the electromagnetic theory says that the increase in intensity of the Raman signal for adsorbed bits on particular surfaces occurs because of the enhancement in the electric field provided by the surface. And when the incident light in the experiment strikes the surface, localized surface plasmons are excited, and the field enhancement is greatest when the plasmon frequency is in resonance with the radiation. And in order for scattering to occur, the plasmon oscillations must be perpendicular to the surface, and if they are in plane with the surface, no scattering will occur. It is because of this requirement that roughened surfaces or arrangements of nanoparticles are typically employed in SIRS experiments, as these surfaces provide an area on which these localized collective oscillations can occur. So SIRS enhancement can occur even when an excited molecule is relatively far apart from the surface, which hosts metallic nanoparticles, enabling surface plasmon phenomena. Okay, so the light incident on the surface can excite a variety of phenomena in the surface, yet the complexity of the situation can be minimized by surfaces with features much smaller than the wavelength of the lights, as only the dipolar contribution will be recognized by the system, and the dipolar term contributes to the plasmon oscillations, which leads to the enhancement. So the SIRS effect is also pronounced because the field enhancement occurs twice. First, the field enhancement occurs. Field enhancement magnifies the intensity of the incident light, which will excite the Raman modes of the molecule being studied, therefore increasing the signal of the Raman scattering. The Raman signal is then further magnified by the surface due to the same mechanism that excited the incident light, resulting in a greater increase in the total output. At each stage, the electric field is enhanced as for a total enhancement. So the enhancement is not exactly equal for all the frequencies. For those frequencies for which the Raman signal is only slightly shifted from the incident light, both the incident laser light and the Raman signal can be near resonance with the plasmon's frequency, leading to the enhancement. When the frequency shift is large, the incident light and the Raman signal cannot both be on resonance with the omega. Thus, the enhancement at both stages cannot be maximal. So the choice of surface material metal is also dictated by the plasmon resonance frequency. So visible and near infrared radiation are used to excite Raman modes. Silver and gold are typical metals for SERS experiments because their plasmon resonance frequencies fall within these wavelength ranges, providing maximal enhancement for visible and NIR light. So the copper's adsorption spectrum also falls within the range acceptable for the SERS experiments. And platinum and palladium nanostructures are also display plasmon resonance within visible and the NIR frequencies. So there's another theory called the chemical theory. So the resonance Raman spectroscopy explains the huge enhancement of Raman scattering intensity, and intermolecular and intramolecular charge transfer significantly enhance Raman spectrum peaks. So in particular, the enhancement is huge for species adsorbing the metal surface due to the high intensity charge transfers from the metal surface with wide band to the adsorbing species. And this resonance Raman enhancement is dominant in SERS for species on small nanoclusters with considerable band gaps because surface plasmon appears only in metal surface with near zero band gaps. And this chemical mechanism probably occurs in concert with the electromagnetic radiation mechanism for metal surface. Okay, so while SERS can be performed in colloidal solutions, today the most common method for performing SERS measurements is by depositing a liquid sample onto a silicon or glass surface with a nanostructured noble metal surface. So while the field experiments were performed on electrochemically roughened silver, now surfaces are often prepared using a disruption of the structure basically. So using basically wait let me yeah so while the first experiments were actually performed on electrochemically roughened silver, now surfaces are often prepared using a distribution of metal nanoparticles on the surface, as well as they can also use lithography or porous silicon as a support. So two-dimensional silicon nanopillars decorated with silver have also been used to create SERS active substrates. And the most common metals used for plasmonic surfaces in visible light, as I said, are silver and gold. However, aluminium has also recently been explored as an alternative plasmonic material because its plasmon band is in the UV region, contrary to silver and gold. So there's a great interest in using aluminium also for UV SERS, and it has however surprisingly also been shown to have a large enhancement in the infrared, which is not fully understood. And in the current decade, it has been recognized that the cost of SERS substrates must be reduced in order to become a more commonly used analytical chemistry measurement technique. So to meet this need, plasmonic paper has experienced widespread attention in the field, with highly sensitive SERS substrates being formed through approaches such as soaking, then in situ synthesis, screen printing, and inkjet printing, and all of that. Okay, so the shape and the size of the metal nanoparticles also strongly affect the strength of the enhancement because these factors influence the ratio of the absorption and the scattering events. And there is an ideal size for these particles and an ideal surface thickness for each experiment. So if concentration and the particle size can be tuned better for each experiment, this will go a very long way in the cost reduction of the substrates. So particles that are too large allow the excitation of multipoles which are non-radiative. As only the dipole transition leads to Raman scattering, the higher order transitions will cause a decrease in the overall efficiency of the enhancement. So particles that are too small lose their electrical conductance and cannot enhance the field. And when the particle size approaches a few atoms, the definition of a plasmon does not hold, as there must be large collection of electrons to oscillate together. An ideal SIRS substrate must possess high uniformity and high field enhancement, and such substrates can be fabricated on wafer scale. And label-free super resolution microscopy has also been demonstrated using the fluctuations of surface enhanced Raman scattering signal on such highly uniform, high performance plasmonic nano surfaces. Okay. So, due to their unique physical and chemical properties, two-dimensional materials have gained significant attention as alternative substrates for surface enhanced Raman scattering spectroscopy. The use of 2D materials as SIRS substrates offers several advantages over traditional metal substrates, including high sensitivity, reproducibility, and chemical stability. So graphene is, I think, one of the most widely studied 2D material for SIRS applications, and graphene has a high surface area, high electron mobility, and excellent chemical stability, making it an attractive substrate for SIRS. So graphene-based SIRS sensors have been shown to be highly reproducible and stable, making them attractive for real-world applications. And in addition to graphene, either 2D materials, especially like mycines, have also been investigated for SIRS applications. And like the applications are one and many. SIRS substrates are used to detect the presence of low abundance biomolecules and can therefore detect proteins in bodily fluids. Early detection of pancreatic cancer biomarkers was accomplished using SIRS-based immunoassay approach. A SIRS-based multiplex protein biomarker detection platform in a microfluidic chip is used to detect, several protein biomarkers to predict the type of disease and critical biomarkers and increase the chance of diagnosis between diseases with similar biomarkers. So this technology has been utilized to detect urea and blood plasma level free in human serum and may become the next generation in cancer detection and screening. So the ability to analyze the composition of a mixture at a nanoscale makes the use of SIRS substrates that are beneficial for environment analysis, pharmaceuticals, material sciences, art and archaeological research, forensic science, drug and explosives detection, food quality analysis, single algal cell detection. So SIRS combined with plasmonic sensing can be used for high sensitivity quantitative analysis of small molecules in human biofluids, the quantitative detection of biomolecular interaction, the detection of low-level cancer biomarkers via sandwich immunoassay platform, the label-free characterization of exosomes, and the study of even redox process at a single molecule level. So that's why SIRS is a very powerful technique for determining structural information about molecular systems. SIRS-based immunoassays are also very important in the detection of low abundance biomarkers. For example, antibodies and gold particles can be used to quantify proteins in serum with high sensitivity and specificity. Okay, so you can also do oligonucleotide targeting with SIRS. SIRS can be used to target specific DNA and RNA sequences using a combination of gold and silver nanoparticles and Raman active dyes such as Cy3. So specific single nucleotide polymorphisms or SNPs can be identified using this technique. So the gold nanoparticles facilitate the formation of a silver coating on the dye-labeled regions of the DNA and RNA, allowing SARS to be performed. So these are several potential applications. For example, there was a paper where they reported that gene sequences for HIV, Ebola, hepatitis and Bacillus anthracis can be uniquely identified using this technique. And each spectrum was specific, which is advantageous over fluorescence detection. Some fluorescent markers overlap and interfere with other gene markers. So the advantage of this technique to identify gene sequences is that several Raman dyes commercially available, which could lead to the development of non-overlapping probes for gene detection. Okay, so our question regarding SIRS was which of these is false. So as Neha said, none of the above, which is the right answer. So that means does SIRS have a very high sensitivity? So yes, SIRS is known to have very high sensitivity. It's a technique that enhances basically the Raman scattering signal by several orders of magnitude by using nanostructured metal surfaces such as silver or gold, basically to amplify the Raman signal of the target molecules. So the sensitivity of SIRS depends on several factors, including the size, shape and composition of the metal nanoparticles, as well as the nature of the target molecules and their interaction with the nanoparticles. However, in general, SIRS can detect even trace amounts of target molecules with high sensitivity and specificity. So SIRS has been used for a wide range of applications, including biomedical diagnostics, environmental monitoring and food safety testing. The SIRS-based biosensors have been developed for the detection of various biomolecules, including proteins, nucleic acids, and viruses with high sensitivity and specificity. So overall, SIRS is a very powerful and highly sensitive analytical technique that has the potential to revolutionize various fields of research and industry. Also, this point is correct. The first option is correct. Does SIRS have very high specificity? Yes. It's this has a very high specificity. The specificity of SIRS depends on several factors, including the size, shape, and the composition of the metal nanoparticles, as well as the nature of the target molecules and their interactions with the nanoparticles. So the unique spectral fingerprint of the Raman signal allows for the identification and the quantification of specific molecules, and SIRS can provide high resolution information about the chemical composition and structure of the target molecules. So SIRS-based biosensors have been developed for the detection of various biomolecules, including proteins, nucleic acids, and viruses with high specificity and sensitivity. For the specific binding of the target molecules to the functionalized metal nanoparticles or surfaces provides high selectivity, allowing for the detection of specific biomarkers even in complex mixtures. So overall, SIRS is a very powerful technique in the world right now. Also, this second option is also correct. Does SIRS use Raman signals? Yes, definitely. It basically Raman spectroscopy is a technique that measures the inelastic scattering of photons by molecules, which provides information about the vibrational modes of the molecules. In SIRS, that is surface enhanced Raman scattering, the Raman signal of the target molecules is enhanced by several orders of magnitude by using nanostructured metal surfaces such as silver or gold to amplify the Raman signal. So the inserts, the target molecules are adsorbed onto the metal surface, and the incident laser light interacts with the metal nanoparticles, causing the formation of localized surface plasmons, and the electric field of the plasmons interacts with the target molecules, enhancing the Raman scattering signal. So the Raman signal generated in SIRS provides high resolution information about the chemical composition and the structure of the target molecules, allowing for their identification and quantification. So SIRS can detect even trace amounts of target molecules with high sensitivity and specificity, making it a very powerful analytical tool for various fields of research and industry. Okay, so overall, SIRS is a type of Raman spectroscopy that enhances the Raman signal of the target molecules by using nanostructured metal surfaces, allowing for the detection and the quantification of specific molecules with high sensitivity and specificity. So yes, all these three options are true. So none of the above options are false. So that was it for this question. We'll move on to the next question. Let me check if I'm still in the meeting. Yeah. Okay. Yeah. So I'm still in the meeting. Moving on to the next question. With regard to COVID-19 pandemic, which of the following is wrong? Is false, basically. So these are the options. Re-emergence of novel coronavirus that caused MERS in 2012. Okay. Caused due to the antimicrobial resistance of common cold causing pathogen, and it's due to a specific mutation in the viral RNA polymerase PB1F2 of H1N1 virus. All of the above, none of the above. Which of these options is true? Like with regard to COVID-19, which is false? So you have to choose that answer which is false. Sorry. Anybody? Anybody? Is anybody saying something? I am not able to hear. You can type it out also in case there is an issue. Okay, so all of the above options are false. I'll explain them one by one. So COVID-19 is basically a disease caused by this novel coronavirus known as SARS-CoV-2, severe acute respiratory syndrome coronavirus 2. So this virus was first identified in Wuhan, China in December 2019 and has since then spread globally, leading to the pandemic. So I don't think I need to explain to anyone what the pandemic was basically, but I'll explain what SARS-CoV-2 is. SARS-CoV-2 is a single-stranded RNA virus that belongs to the family Coronaviridae. It is a member of the subfamily Orthocoronavirinae which also includes the viruses that caused the SARS outbreak in 2002-2003 and the MERS outbreak in 2012. MERS full form is Middle East Respiratory Syndrome. So their subfamily is same. But SARS-CoV-2 is a new strain of coronavirus that has not been previously identified in humans. So the virus is thought to have originated in bats and then been transmitted to humans, possibly through an intermediate host such as a pangolin. So SARS-CoV-2 primarily spreads through respiratory droplets when an infected person, talks or coughs or sneezes. And the virus can also be transmitted by touching contaminated surfaces and then touching one's face. Okay, so the disease caused by SARS-CoV-2 is COVID-19. So the symptoms of COVID-19 can range from mild to severe, with some people being completely asymptomatic. So common symptoms include fever, cough, fatigue, difficulty breathing, and in severe cases, COVID-19 can actually lead to pneumonia, acute respiratory distress syndrome, and even death. So the incubation period for this SARS-CoV-2 ranges from 2 to 14 days with an average of 5 days, and the severity of COVID-19 is influenced by various factors including age, underlying health conditions and immune system response. So older adults and those with underlying health conditions such as diabetes, heart disease and lung disease are at a higher risk of developing severe COVID-19. Okay, so what are the prevention and the treatment steps for COVID-19? There is currently no specific treatment for COVID-19, like nothing to treat as such. We have vaccines that will be injected in your body that can basically try to subdue it. Most people with mild symptoms recover at home only with self-care measures such as rest, hydration and over-the-counter medications to reduce fever and pain. In severe cases, hospitalization might be necessary to provide, supportive care such as oxygen therapy, mechanical ventilation and other treatments to manage, complications. Otherwise, people are now getting vaccinated, so there's a also many people, I think most people have been infected by the virus at least once. So that also has created herd immunity. So also prevention measures, we all know: wearing masks, practicing physical distancing, washing hands frequently with soap and water, avoiding crowded places and large gatherings. So vaccines are also available to prevent COVID-19, and vaccination is recommended for eligible individuals to reduce the spread of the virus and prevent severe illness and death. So in conclusion, COVID-19 is a respiratory illness caused by this novel coronavirus SARS-CoV-2, and the virus primarily spreads through respiratory droplets and can lead to mild to severe symptoms, with older adults and those with underlying health conditions being at a higher risk of severe illness. And prevention measures, obviously we all know: masks, physical distancing, vaccinations, hand hygienes are recommended to reduce the spread of the virus and prevent severe illness and death. So what was this option? So is COVID-19 a re-emergence of the novel coronavirus that caused MERS in 2012? So no. COVID-19 is not a re-emergence of the novel coronavirus that caused Middle East Respiratory Syndrome or MERS in 2012. While both COVID-19 and MERS are caused by coronaviruses, they are caused by different viruses with distinct genetic sequences and different clinical manifestations. The virus that causes COVID-19 is known as SARS-CoV-2 or Severe Acute Respiratory Syndrome Coronavirus 2, while the virus that causes MERS is known as MERS-CoV, which is Middle East Respiratory Syndrome Coronavirus. So both these viruses are zoonotic, meaning they were transmitted to humans from animals, but they have different animal reservoirs. MERS-CoV is believed to have originated in dromedary camels, while the origins of SARS-CoV-2 are still being studied but are thought to be linked to bats. So while COVID-19 and MERS share some similarities in their clinical manifestations such as fever, cough, and respiratory symptoms, they also have important differences. MERS is generally more severe than COVID-19 with a higher mortality rate, but it is comparatively less contagious than COVID-19. COVID-19 has a higher transmission rate and can be spread by even asymptomatic individuals, while MERS is primarily transmitted through close contact with infected individuals in healthcare settings. So, in summary, COVID-19 and MERS are caused by different viruses belonging to the same subfamily but still different viruses with distinct genetic sequences and different clinical manifestations, and while they share some similarities, they are not the same virus. So, the first option is wrong. Is COVID-19 pandemic caused due to antimicrobial resistance of common cold causing pathogen? No. COVID-19 pandemic is not caused by antimicrobial resistance of the common cold causing pathogen. It is caused by SARS-CoV-2, which is different from the viruses that cause the common cold. Antimicrobial resistance, or AMR, refers to the ability of microorganisms such as bacteria, viruses, fungi, and parasites to resist the effects of antimicrobial drugs such as antibiotics, antivirals, and antifungals. Okay. So while AMR, or antimicrobial resistance, is a global public health threat, it is not the cause of COVID-19. The spread of COVID-19 is primarily due to the person-to-person transmission through respiratory droplets when an infected person talks, coughs, or sneezes. Virus can also be transmitted by touching contaminated surfaces and then touching one's face. So while the common cold is caused by various viruses including rhinoviruses, coronaviruses, and adenoviruses, the viruses that cause the common cold are not responsible for the COVID-19 pandemic. COVID-19 is caused by a novel coronavirus that was first identified in Wuhan, China in December 2019. So in summary, COVID-19 pandemic is caused by a novel coronavirus called SARS-CoV-2 and is not related to the antimicrobial resistance of the common cold causing pathogen. Okay. So second option is also incorrect. So what about the third option? Is COVID-19 due to a specific mutation in the viral RNA polymerase PB1F2 of H1N1 virus? So no. This is COVID-19 is not due to a specific mutation in the viral RNA polymerase PB1F2 of H1N1 virus. It is caused by SARS-CoV-2 severe acute respiratory syndrome coronavirus 2, which is a different virus from the H1N1 influenza virus that caused the 2009 influenza pandemic. So the PB1F2 protein is a virulence factor produced by the influenza A virus, including the H1N1 strain that caused the 2009 pandemic. So some studies have suggested that certain mutations in the PB1F2 protein may be associated with increased virulence and severity of the H1N1 virus. However, these mutations are in no way related to the SARS-CoV-2 virus that causes COVID-19. The genetic sequence of SARS-CoV-2 is different from that of the H1N1 influenza virus, and the mechanisms of infection and the pathogenesis of these viruses are also very different. Okay, SARS-CoV-2 primarily targets the respiratory system, while influenza viruses can also affect other systems of the body. So in summary, COVID-19 is not due to the specific mutation in the viral RNA polymerase PB1F2 of H1N1 virus. COVID-19 is caused by a novel coronavirus called SARS-CoV-2, which is a different virus from the H1N1 influenza virus. Okay, so all these three options are false. That is why the answer will be all of the above, because the question is asked with regard to COVID-19 pandemic, which is false. So all of the above are false. Okay. Moving on to the next question. Biosensors consist of bioreceptors plus transducers, enzymes as bioreceptors plus transducers, both, none. Which do you think is the correct option? Anyone? Is anyone saying something? I'm not able to hear. You can type also if I can hear that someone has unmuted, but I'm not able to hear what you're saying. Okay, I'm not able to hear. If you're typing something, I'll check. Also, you can see the answer anyway. Yes, correct, both. Yeah. So we know that biosensors are obviously devices that detect and quantify biological or biochemical analytes, and they typically consist of three parts: the bioreceptor, transducer, and the signal processor. Okay. So bioreceptor is a biological component of the biosensor that interacts with the target analyte. It is typically an enzyme antibody or nucleic acid that specifically recognizes and binds to the analyte. Okay, for example, in a glucose biosensor, the bioreceptor is glucose oxidase, which specifically reacts with glucose. The next component is the transducer. So the transducer is the component of the biosensor that converts the biological signal from the bioreceptor into a measurable signal. So this can be done by various means, including electrical, optical, or mechanical signals. For example, in an electrochemical biosensor, the transducer is an electrode that converts the biochemical reaction between the analyte and the bioreceptor into an electrical signal. Then we have the signal processor. The signal processor is the component of the biosensor that receives the signal from the transducer and converts it into a readable output. And this can be done using various methods, including digital signal processing or analog circuitry. For example, in a glucose biosensor, the signal processor converts the electrical signal generated by the transducer into a glucose concentration reading. Okay. So overall, the bioreceptor, transducer, and signal processor work together to detect and quantify analytes in a sample. The specificity and the sensitivity of the biosensor depend on the quality and the properties of each component, okay, as well as the overall design of the biosensor. So biosensors have various applications in healthcare, environmental monitoring, food safety, and many other fields. So can enzymes act as, so what was the question? Enzymes as bioreceptors. Can enzymes act as bioreceptors? Yes, enzymes can very well act as bioreceptors in biosensors. In fact, enzymes are most commonly used as bioreceptors in biosensors due to their high specificity and sensitivity for their target analytes. Okay. Enzymes catalyze biochemical reactions that are specific to their substrate, and this specificity can be used to selectively detect and quantify target analytes. For example, glucose oxidase can be used as a bioreceptor in a biosensor to detect glucose levels in a sample. And the enzyme specifically reacts with glucose, producing hydrogen peroxide and glutonic acid as byproduct. Okay, and this reaction can be coupled with the transducer to generate a measurable signal such as electric current or change in color, which can be processed to provide a glucose concentration reading. Other examples of enzymes that can be used as bioreceptors in biosensors include lactate dehydrogenase, which can be used to detect lactate levels in acetylcholinesterase, which can be used to detect pesticides. So enzyme-based biosensors have applications in medical diagnostics, environmental monitoring, and food safety, among other areas. So what else can be a bioreceptor except enzymes? In addition to enzymes, other types of biomolecules can act as bioreceptors in biosensors, which includes antibodies. Okay, so antibodies are proteins produced by the immune system that specifically recognize and bind to foreign substances known as antigens. Antibodies can be used as bioreceptors in biosensors to detect a wide range of analytes, including bacteria, viruses, and proteins. Nucleic acids. Nucleic acids such as DNA and RNA can also act as bioreceptors in biosensors. They can be designed to specifically recognize and bind to complementary DNA and RNA sequences, allowing them to detect specific genetic targets. Receptors. Receptors are proteins that bind to specific signaling molecules such as hormones or neurotransmitters, and they can be basically used as bioreceptors in biosensors to detect these molecules in biological samples. Or even microorganisms. So whole cells or microorganisms can also be used as bioreceptors in biosensors. They can be genetically engineered or selected for their ability to specifically interact with the target analyte, allowing them to detect the presence of the analyte in the sample. So overall, the choice of bioreceptor depends on the specific analyte being detected and the characteristics of the sample matrix. The bioreceptor must be selected for its high specificity and sensitivity to the analyte of interest, as well as its stability and reproducibility in the biosensor. Okay. So both of these questions are correct. Okay. So yes, biosensors can be classified on a lot of factors. The classification of biosensors can happen based on the bioreceptors also. Okay, so according to the biorecognition principle, as I said, biosensors can be classified as catalytic biosensors or non-catalytic biosensors. Okay, in a catalytic biosensor, analyte bioreceptor interaction results in the development of a new biochemical reaction product, and this biosensor includes enzymes, microorganisms, tissues, and whole cells. In the case of affinity or non-catalytic biosensor, the analyte is bound to the receptor irreversibly, and during this interaction no new biochemical reaction product is formed. This type of sensor comprises antibodies, cell receptors, and nucleic acids as a target for detection. So the most common is the enzymes based biosensors. So enzymes are common biocatalysts which are efficient at increasing the biological reaction rate, and the working principle of an enzyme based biosensor depends on the catalytic reaction and the binding capabilities for the target analyte detection, and various possible mechanisms are involved in the analyte recognition process. So the analyte is metabolized by the enzyme. So the enzyme concentration is estimated by measuring the catalytic transformation of the analyte by the enzyme. Also, an enzyme inhibited or activated by the analyte. So the analyte concentration is related to the decreased enzymatic product formation, and also tracking of the alteration of the enzyme characteristics. And owing to the long history of enzyme-based biosensors, various biosensors can be produced on the basis of enzyme specificity. However, the enzyme structure is extremely sensitive, which makes it expensive and complicated to improve its sensitivity, stability, and adaptability. Okay, so electrochemical transducers are most commonly used for enzyme-based biosensors. The most common enzyme-based biosensors are glucose and urea biosensors. Okay, so Cordero et al. Developed and characterized some amperometric enzyme-based glucose biosensors for real-time monitoring of glucose in the brain in vitro, and the experiments reveal that developed biosensors can monitor changes in brain glucose in response to relevant pharmacological challenges. So there was another author, some Uygun et al. Developed a highly stable potentiometric urea biosensor using nanoparticles. So the response time and detection limit of the developed sensor was, I think, 30 seconds and 0.77 micromolar respectively. So integrating enzymes with nanomaterials has resulted in increased use of enzymes as recognition elements in biosensors. There can be antibody-based biosensors as well. So antibodies are affinity biorecognition elements which have been used over 2 decades almost because of their wide application range and strong antigen-antibody interactions. Antibodies possess the structure of immunoglobulins in the form of the Y shape, which consists of 2 heavy and 2 light polypeptidic chains connected by disulfide bonds. So 5 classes of antibodies have been identified based on the differences in heavy chains: IgG, IgM, IgA, IgT, and IgE. So biosensors that have an embedded antibody as ligands or function on the antibody-antigen interaction are called immunosensors, and immunosensors are classified as non-labeled or labeled. Okay, so non-labeled immunosensors are constructed to specifically determine the antigen-antibody complex by estimating the physical changes caused by the development of the complex. In the case of labeled immunosensor, a sensitivity detectable label is introduced. So the antigen-antibody complex is sensitively assessed through label measurement. So Maduro et al. Constructed a label-free immunosensor to detect ovarian cancer. Okay, the system has a linear relationship of anti-CA125 concentration in the range of 5 to 80 unit per ml, exhibiting a limit of detection of 1.45 unit per ml. And there was another called Marquette et al. Who developed electrical and optical biosensing for label-free detection of aflatoxin B1 using gold nanobipyramids, surface plasmon resonance based AFP aflatoxin. Detection was found to be in the linear range of 0.51 to 500 nanomolar with a detection limit of 0.4 nanomolar, while impedimetric aflatoxin detection was exhibited in the linear range of 0.1 to 25 nanomolar, okay, having a detection limit of 0.1 nanomolar. They can be aptamer based biosensors as well. So aptamers are synthetic single-stranded nucleic acids that are sequences of DNA or RNA that bind to target molecules selectively and can be folded into 2-dimensional and 3-dimensional structures. So in 2D or 3D structures, the targets have high binding performance due to greater surface density and less spatial blocking. So due to the nucleic acid character of aptamers, they are structurally and functionally stable over a wide range of temperatures and storage conditions. Unlike antibodies that require biological systems to be generated, aptamers can be chemically synthesized, are stable in a pH range of 2 to 12, and have certain thermal refolding capabilities. A further benefit of aptamers is that they can be chemically modified according to the detection criteria for the target molecule. So by an in vitro selection system, selects or systemic evolution of ligands by exponential enrichment. Aptamers can be isolated from oligonucleotides libraries, and several selects variants have been recently established, including cell selects, capillary electrophoresis based selects, microfluidic selects, fax based selects, microtiter plate selects, magnetic bead selects, and in vivo selects. So optical, electrochemical, and piezoelectric techniques are the most frequently used in biosensors. Depending on different transduction techniques, these biosensors are further categorized as labeled or label-free aptasensors. So surface plasmon resonance or SPR is the most commonly used method for the label-free optical sensors, whereas fluorescent dyes fluorescent are used for label-based optical aptasensors. For tracking biological systems in real time, fluorescent nanoparticles such as QDs provides many benefits over regular fluorescent dyes. To identify targets such as cancer cells, bacterial spores, and proteins, aptamer QD conjugates can be used, and there have been like more biosensors like for thrombin protein and etc. that have been published in papers. If you're interested, you can actually go online and read more about it. Then we come to whole cell biosensors. So whole cell based biosensors utilize microbes such as bacterial, fungi, yeast molds, algae, protozoa, and viruses, since they possess potential biorecognition elements in the construction of whole cell based biosensors. So they are self replicating and can produce recognition elements such as antibodies without the need for extraction and purification. So compared with animal or plant cells, whole cell based biosensors are easy to handle and rapidly proliferating. The cells can interact with a wide variety of analytes, display the electrochemical response that a transducer can register, and can transmit whole cell based biosensor principle. So owing to their good sensitivity, high selectivity, and capability of detection, these biosensors were successfully employed in environmental monitoring, food analysis, pharmacology, heavy metals, pesticides, detection of organic contaminants, and drug screening. So Polizzi et al. Actually developed label-free optical whole cell Escherichia coli or E. coli biosensors to detect pyrethroid insecticide exposure with a detection limit of 3 nanogram per ml, 3 ppm in the linear range of 0.01 to 2 nanogram per ml. Lastly, we can also find nanoparticle based biosensors. So apart from the above classes of bioreceptors, nanoparticles or nanomaterials have recently been considered as a new category of bioreceptors. So with the advancement of nanotechnology and nanoscience, different nanoparticles have been used as bioreceptor. So nanoparticles deliver a more vast range of applications in biosensing technology, and these can act as bioreceptors. These can act as bioreceptors as well as transducers. For example, cerium oxide based nanoparticles exhibit biomimetic catalytic activity favorable for bioreceptors. So due to their effective transduction capabilities, different inorganic materials such as graphene and CNTs based nanomaterials, noble metal nanoparticles, and QDs have successfully been used as transducers. So these are some of the most commonly used bioreceptors. So yes, this option in this question both are correct. Moving on to the next question. Piezoelectric biosensors use platinum to detect the specific frequency at which photons are emitted when the substance is exposed to magnetic field. Is it true or false? Anyone? Okay, false. Yes, you're correct. It's false. So, piezoelectricity or piezoelectric effect is basically a physical phenomenon which refers to an ability of a material to produce voltage when mechanically stressed. The effect works in the opposite situation as well. Voltage given to the surface of a piezoelectric material causes mechanical stress or oscillation when the voltage is alternating. So anisotropic crystals, that is crystals without center of symmetry, or are typical materials exerting piezoelectricity. This is aluminum phosphate, aluminum nitride, zinc oxide, crystallized topaz, barium and lead titanate, gallium orthophosphate, quartz, tartrate tetrahydrate, polyvinylidene fluoride, polylactic acids can be these can be mentioned as typical piezoelectric materials. So considering an analytical chemistry point of view, the piezoelectricity is well suitable for physical sensors and biosensors construction. And the principle of such assay can be introduced by following a very simple explanation. The biosensor or sensor is excited by alternating voltage given on the surface of two electrodes. The alternating voltage causes mechanical oscillations of crystal, and frequency of oscillations is measured as the crystal is put into the oscillation circuit. Analyte or any other mass bound on surface of crystal, or more precisely on the surface of electrodes located on the crystal, results in the change in the oscillation frequency, and the change or the decay in the frequency is proportional to the mass bound. The crystal as described in a paper by Sauverbray. I'm sorry if I'm not able to pronounce the name properly. Sauverbray and co-workers. But change in the medium viscosity can also influence the oscillations as defined by Kanazawa and his co-workers. Kanazawa and their co-workers. So the piezoelectric platform appears to be ideal for the construction of biosensors. It can simply record affinity interactions without the necessity to apply any specific reagents. On the other hand, sensitivity in microorganisms necessary to make measurable change in oscillations and some specific aspects like fragility should also be taken into consideration. So the number of piezoelectric platforms is quite wide, and many papers on this issue have been published. And as the number of adaptations is high, the number of used biorecognition paths and designs of the biosensors is high as well. So particular types of biorecognition paths of piezoelectric biosensors are explained in some review papers online if you want to check it out. Basically, they cover the recent development in this field. That is what they do. So what are piezoelectric immunosensors? Piezoelectric immunosensors are analytical devices which can be performed for the basically for the determination of various macromolecular compounds in microorganisms. The piezoelectric immunosensors are biosensors which contains an antibody as a biorecognition element, and specificity of the antibody significantly influences the specificity of the whole immunosensor. So in a general expression, a piezoelectric immunosensor is specific when the used antibody is specific, and the electrode with the other sensitive parts of the piezoelectric material will not be sensitive to the unspecific reactions with interfering compounds. So though immunosensors typically contain immobilized antibody and they are able to recognize antigens, the opposed reaction is possible as well. It means that the immunosensor can contain immobilized antigen and be used for the recognition of an antibody, and just antibody is the analyzed molecule. So the piezoelectric immunosensor with an immobilized antigen are therefore suitable tool for diagnosis of infectious diseases. Okay, so this is the principle of the piezoelectric immunosensor. So the statement that is written here is not entirely accurate. Piezoelectric biosensors do not typically use platinum to detect the specific frequency at which photons are emitted when the substance is exposed to a magnetic field. So piezoelectric biosensors rely on the piezoelectric effect, which is the ability of certain materials such as quartz or ceramics to generate an electrical charge in response to mechanical stress. So when a piezoelectric material is mechanically stressed, it produces a voltage difference across its surface, which can be measured and correlated to the amount of stress applied. So in a piezoelectric biosensor, the bioreceptor such as an antibody enzyme is immobilized onto the surface of the piezoelectric material. So when the target analyte binds to the bioreceptor, it causes a change in the mechanical stress of the piezoelectric material, which results in a measurable change in the voltage output. So this change in the voltage output can be recorded and correlated to the concentration of the target analyte in the sample. Platinum may be used in some piezoelectric biosensors as an electrode to facilitate the measurement of the voltage output. However, platinum is not directly involved in the detection mechanism of the biosensor. Okay, so that is why this statement is not true, it's false. Okay, because this statement is false because it confuses the technology of piezoelectric biosensors with that of other types of sensors and detectors. Okay, the bioreceptor such as an antibody or enzyme is immobilized onto the surface of the piezoelectric material. So when the target analyte binds to the bioreceptor, it causes a change in the mechanical stress on the piezoelectric material, which results in a measurable change in the voltage output. So this change in voltage can be recorded and correlated to the concentration of the target analyte in the sample. In contrast, the statement mentions that the detection of a specific frequency of photons emitted when the substance is exposed to a magnetic field. This type of detection, it involves a completely different mechanism such as fluorescence, spectroscopy, or photoluminescence, which are not related to the piezoelectric effect also. So therefore, the statement is not correct, it's false. Yes, it's false. So she was correct. Yeah, it's false. Moving on to the next question. Which of the following is not a commonly used bioinformatics tool for understanding biological data? Megan, Adobe Photoshop, Adobe Photoshop, Kine, Blast. Which one do you think is not a commonly used bioinformatics tools for understanding biological data? Anyone? Are you saying anything? B. Yes, correct. C. Yes, Dr. Arvind is correct. B is not commonly used with bioinformatics software. So Megan is called Metagenome Analyzer. It is a bioinformatics tool designed for analyzing large metagenomic data sets. Okay, data sets, which are collections of genetic sequences from complex microbial communities. So this tool was developed by Daniel Husson and his team at the University of Tübingen in Germany. So, Megan uses a variety of algorithms to analyze and visualize metagenomic data, including BLAST and the lowest common ancestor algorithm. So, these algorithms allow Megan to classify metagenomic reads based on their similarity to known sequences in the public databases such as GenBank or NCBI. Megan can also perform functional analysis of metagenomic data, allowing researchers to identify genes and metabolic pathways that are present in the microbial community. So it provides several visualization tools, including interactive heat maps, hierarchical clustering trees, and taxonomic sunburst plots, and that allow users to basically explore and interpret their data. Overall, Megan is a powerful tool for analyzing and interpreting metagenomic datasets, providing insights into the taxonomic and functional diversity of complex microbial communities. It is widely used in research areas such as environmental microbiology, human microbiome research, and microbial ecology. Then we move to KIME. KIME is known as Quantitative Insights into Microbial Ecology. So, this is a bioinformatics tool designed for analyzing and interpreting large microbiome datasets. It was developed by Rob Knight and his team at the University of Colorado in 2010 and has since undergone several updates and improvements. So, KIME is specifically designed for analyzing high throughput sequencing data such as 16SR RNA gene sequencing, shotgun metagenomics, and other sequencing technologies, and it provides a suite of tools for quality control, sequence processing, taxonomic classification, and statistical analysis of microbiome data. And KIME is built on a flexible and modular framework, allowing users to choose the specific analysis pipelines and parameters that best suit their research needs. So it includes tools for alpha and beta diversity analysis, differential abundance testing, network analysis, and other advanced statistical analysis. And KIME also provides several visualization tools such as heat maps, rare fraction curves, and like principal PCOA plots to help users explore and interpret the data. In addition, it offers extensive documentation and support, making it accessible to researchers with varying levels of bioinformatics expertise. Overall, KIME is a powerful and widely used tool for analyzing and interpreting microbiome data. It has been used in a wide range of research areas such as human microbiome research, environmental microbiology, and microbial ecology, and has contributed significantly to our understanding of the role of microbiomes in health and disease. Then we move to BLAST. I think you must have heard about BLAST. BLAST is a basic local alignment search tool, which is widely used by informatics tool for comparing nucleotide or protein sequences to databases of the known sequences. Even I have used BLAST. It was developed by Stefan Alschul and the colleagues at the National Institute of Health (NIH) in the 1990s, and has since become an essential tool for molecular biology and genetics research. So BLAST uses a scoring system to evaluate the quality of the alignment, with higher scores indicating higher levels of similarity. The tool provides several options for customizing the search parameters, including specifying the type of sequence being searched, be it nucleotide or protein, the database to be searched, and the scoring matrix used to evaluate the alignment. So BLAST output includes a list of the most similar sequences found in the database, along with the alignment scores and other relevant information. And users can further analyze the results, such as identifying conserved domains or designing primers for amplification. Overall, BLAST is a powerful and widely used tool for molecular biology and genetic research, providing researchers with a quick and efficient way to identify homologous sequences and infer functional relationships between proteins and genes. Next, we move on to Adobe Photoshop. Adobe Photoshop is a software application used for image editing and graphic design. It was first developed in 1988 by Thomas and John Knoll and has since become the industry standard for digital image manipulation. So, Photoshop is a versatile tool that allows users to create and edit images, graphics, and illustrations. Its features include advanced color correction, image retouching, and composition tools, as well as 3D design capabilities and video editing tools. Users can edit images in Photoshop using a variety of techniques, including cropping, resizing, and adjusting brightness and contrast. So, they can also use the software to apply filters and effects, add text, draw shapes, and lines. And Photoshop uses layers to organize and manipulate images. So each layer can contain a separate image or graphic element which can be edited independently of the other layers. So this allows users to create complex designs and compose multiple images together. Sorry. Photoshop also provides a range of selection tools allowing users to isolate specific areas of an image or for editing. And this includes the lasso tool, the image magic wand tool, and the quick selection tool. So overall, Adobe Photoshop is a powerful and widely used tool for image editing and graphic design, used by photographers, graphic designers, and digital artists alike, and it has become an essential tool in the creative industry, enabling users to bring their ideas to life and create stunning visual designs. So yes, so Adobe Photoshop is not a commonly used bioinformatics tool for understanding biological data. So yes, that is why this option should be excluded. So yeah, Arvind was right. Adobe Photoshop. Moving on to the next question. How does sequencing depth affect the accuracy of a sequencing run? And these are your options. Anyone? Anyone? So over the past decade, the field. Yes, anybody is answering? A4. Okay. See the options again. So I'll explain something first. So over the past decade, the field of metagenomics has enabled substantial advancement in the knowledge of microbial ecology, evolution and diversity. And this includes a more in-depth understanding of complex microbial communities within the gastrointestinal tract of animals and humans also. So culture-independent metagenomic approaches can aid in understanding the ecological role, phylogeny, and functionality of gut microbial communities in relation to host physiology. So, metagenomic shotgun sequencing investigations have been used to reveal specific features of the resistome, mobilome, virulome, and virome. And the increasing prevalence of the antimicrobial resistance in bacteria is one of the most important challenges facing public health. Okay, and it has been proposed that livestock production systems may contribute to an increased prevalence of antimicrobial resistant bacteria and genes in the environment and therefore pose a risk to human health. So globally, more than 57 million kilograms of antibiotics are used annually in food animal production, which may select for antibiotic resistant bacteria that persist through the meat and milk production chain. An investigation of the microbiome and the resistome of farm animals and their environment may provide valuable data and models to estimate the public health risk of antibiotic resistant human infections associated with antibiotic use in food animals. So the reason for saying all this is because metagenomic shotgun sequence analysis are accomplished by unrestricted sequencing of the genomes of all microorganisms present in a sample, including uncultured microorganisms. So fully capturing all DNA sequences originating from every microorganism in a complex environment is impractical despite continuous improvements in sequencing depth. So estimating the required sequencing depth needed to characterize a particular microbiome is important to fulfill the objectives of a given study, such as understanding the ecology of antibiotic-resistant genes and bacteria in food production systems. However, the relatively high cost of metagenomic sequencing can still limit the amount of sequenced reads that can be generated, impacting the biological interpretation of resulting data. So, the answer will be 4. Accuracy increases initially with the increase of sequence depth, but eventually reaches the plateau. So, before that, let's learn what sequencing depth actually means. So sequencing depth basically refers to the number of times each base in a genome is read during a sequencing run. So in general, higher sequencing depth can improve the accuracy of a sequencing run because it helps to reduce the errors. Okay, errors caused by sequencing noise such as base calling errors or misaligned reads. So when a base is read multiple times, the consensus sequence can be determined by taking the most frequent base call at each position. And this helps to reduce the impact of sequencing errors or bias, resulting in a more accurate consensus sequence. However, sequencing depth can also have certain limitations. In some cases, extremely high sequencing depth may not significantly improve accuracy and may instead increase the cost and time required for sequencing analysis. Additionally, sequencing depth must be balanced with other factors such as sequencing quality and coverage uniformity. So uneven coverage or low quality sequencing reads can still result in errors even at high sequencing depths. So overall sequencing depth is an important consideration when designing a sequencing experiment and should be balanced with other factors also to achieve the most accurate and cost effective results. So how does the sequencing depth affect the accuracy of a sequencing run? So in like as I said, sequencing depth refers to the number of times each base in a genome is read during a sequencing run. So in general, higher sequencing depth can improve the accuracy of a sequencing run because it helps to reduce the errors caused by sequencing noise. Okay, but as I also said that it also has some limitations. So like in order to get overall sequencing depth will be an important consideration, but it should be balanced. So this first statement says that accuracy increases with the increase of sequence depth. So in general, this statement is can be true that when a base is read multiple times, errors caused by sequencing noise such as base calling errors or misaligned errors can be reduced. So with higher depth, consensus sequences can be determined with greater accuracy. However, there is a limitation to the benefit of increasing sequencing depth. Extremely high sequencing depth may not necessarily improve accuracy and may increase the cost and time required for sequencing analysis. Okay, so as I said before, also sequencing depth should be balanced with other factors such as sequencing quality and coverage uniformity, as uneven coverage or low quality sequencing reads can also result in errors even at high sequencing depths. Okay, so therefore, while it's generally true that increasing sequence depth can increase accuracy, it is important to consider various factors when determining the optical sequencing depth for a particular experiment. The second statement says the accuracy decreases with increasing sequence depth. This is not true. In fact, increasing sequencing depth can often improve the accuracy of sequencing results, especially for complex or highly variable genomes. Okay, um, so this so as I said, when a base is read multiple times, consensus sequences can be generated that reduce the impact of sequencing errors, improving accuracy. However, this assumes that sequencing quality is high and that errors are random rather than systematic. There are some situations where increasing sequence depth may not improve accuracy or may even decrease it. For example, if the genome being sequenced has very low complexity, increasing sequencing depth may not provide additional information, it will just increase the cost and time required for sequencing analysis. Additionally, increasing sequence depth may not be helpful if the sequencing platform or library preparation method introduces systematic errors. So overall, this relationship between sequence depth and accuracy is very complicated and depends on many factors, including the quality of the sequencing platform, library preparation method, and the complexity of the genome being sequenced. However, this statement that accuracy decreases with increase of sequence depth is not true. It's false. Okay. The third statement is there is no relationship between sequencing depth and accuracy. Also, this statement is again false. There is typically a positive relationship between sequencing depth and accuracy up to a certain point. Okay, as I said, in general, sequencing depth increasing can improve accuracy because, in general, increasing sequence depth can improve accuracy because it reduces the impact of sequencing errors and enables consensus sequence generation. However, this also assumes that sequencing quality is high and the errors are random rather than systematic. So there may be diminishing returns to increasing sequencing depth beyond a certain point. Okay, and excessively high sequencing depth may will anyway increase the cost and time required for sequencing and analysis without providing significant improvement in accuracy. So therefore, while it's not accurate to say that there is no relationship between sequencing depth and accuracy, it's still important to consider other factors such as sequencing quality and coverage uniformity and determining the optimal sequencing depth for a particular experiment. Okay. Accuracy, so this statement is also false. So last statement, accuracy increases initially with the increase of sequence depth but eventually reaches its plateau. So this statement is true, generally true. So increasing sequencing depth will improve accuracy by reducing the impact of sequencing errors and enabling consensus sequence generation. However, the benefit of increasing sequencing depth eventually reaches a plateau where additional sequencing reads do not provide a significant improvement in accuracy. Okay, so the point at which sequencing depth reaches the plateau of accuracy depends on the complexity of the genome being sequenced, the quality of the sequencing platform, and the sequencing library preparation method. However, in general, studies have shown that increasing sequencing depth beyond a certain point results in a diminishing return on investment in terms of accuracy improvement. Therefore, it's important to carefully consider the optimal sequencing depth for a particular experiment, taking into account factors such as sequencing quality, coverage uniformity, and the cost and time required for sequencing and analysis. Okay, so yes, the last option is true. So this is a basic sequencing schematic that I have added for a better reference. Okay. So this is the next question. What is the advantage of using a microarray or multiplex PCRs over the traditional PCR methods for detecting microbial communities? So these are your four options. Which do you think is correct? Which one do you think is the correct answer? Okay, someone is typing something. Okay, B3. So there's a confusion between B and C, right? Let's check the options again. Okay. Okay. So the advantage of using microarray or multiplex PCR methods over traditional PCR methods for sure for detecting microbial communities is that they allow for the simultaneous detection and identification of multiple microbial species or strains in a single reaction. You can understand from the name only, it's multiplexed. Okay, so traditional, so the answer is C. You're right, correct. So traditional PCR methods typically amplify a single target sequence, making it really difficult to detect multiple species in a single reaction. In contrast, microarray and multiplex PCR methods use multiple primers or probes to amplify and detect multiple target sequences simultaneously, increasing the throughput and reducing the time and cost of analysis. Okay, and microarray based detection methods use a solid support such as a glass slide or a bead to which multiple probes are attached that can bind to specific target sequences. And the sample DNA is then labeled with a fluorescent dye and hybridized to the microarray, allowing for the detection and the quantification of multiple target sequences simultaneously. So multiplex PCR methods use multiple primer sets in a single reaction, okay, to amplify multiple target sequences simultaneously. The resulting amplicons are then separated and detected using techniques such as gel electrophoresis or high throughput sequencing. Overall, microarray and multiplexed PCR methods offer advantages in terms of speed, sensitivity, and specificity compared to traditional PCR methods for detecting microbial communities. So in that case, speed can also be kind of no. I won't say that multiplex PCRs are speedier than traditional PCR methods, but since in multiplex PCRs you can actually do with a simultaneously detect a larger number of you know uh, basically you can do simultaneously, you can detect multiple microbial species in a single reaction, and while in normal traditional PCR methods it's only one. So in that respect, obviously you're getting everything done at once, and this is in traditional PCR it's only one. Okay, so that's how this might be speedier also, but this option C is a more relevant answer. Okay, so microarray and multiplex PCRs are very related techniques that allow for the detection and identification of multiple genetic targets simultaneously, and these methods are nowadays widely being used in various fields including genomics, microbiology, and diagnostics. And as I said, that microarrays are solid supports that can be made of glass, nylon, or silicon and are coated with probes basically that are complementary complementary to specific target sequences, and the probes are typically short oligonucleotides or DNA fragments that are immobilized onto the surface of the microarray in a precise array pattern, and the sample DNA is then labeled with a fluorescent or radioactive tag and hybridized to the microarray, allowing the target sequences to bind to their complementary probes on the microarray surface. And the microarray is then scanned, and the fluorescent signal intensity is used to quantify the amount of target DNA present in the sample. And multiplex PCR, on the other hand, uses multiple primer sets in a single reaction to amplify multiple target sequences simultaneously. And the primers are designed to recognize unique sequences within the target DNA and amplify them specifically. The resulting amplicons are then separated and detected using techniques such as gel electrophoresis or high throughput sequencing. And multiplex PCRs can be performed using traditional PCR instruments and protocols, making it a very convenient, a relatively easy and cost effective method for detecting multiple genetic targets. So both microarray and multiplex PCR have advantages over traditional PCR methods for detecting microbial communities. Traditional PCR typically uses a single set of primers to amplify a single target sequence, which limits its ability to detect multiple species or strains simultaneously. In contrast, microarray and multiplex PCR allow for simultaneous detection of multiple targets, enabling the analysis of complex microbial communities in a single experiment. So this makes these methods powerful tools for studying microbial ecology, identifying pathogens, and monitoring microbial communities in environmental samples of clinical specimens. Okay, so that's how multiplex PCR is all about. That's how multiplex PCRs are more convenient than normal traditional PCRs. Also, multiplex PCR has been used in pathogen identification, high throughput single nucleotide polymorphism genotyping, high throughput single nucleotide polymorphism genotyping, mutation analysis, gene deletion analysis, template quantification, linkage analysis, RNA detection and forensic studies, and the advantages are many, like internal controls. That you have internal controls, so false negatives due to reaction failure are often shown in multiplex assays because each amplicon provides an internal control for the other amplicons. Also efficiency, it saves preparation time, costly polymerase and templates. Nevertheless, the multiplex PCR has several disadvantages also, mainly including a complex system with many primers, a low amplification efficiency, and no identical efficiency on different templates. So that is something that is an advantage for disadvantage for multiplex PCR. Okay, so that's, but still for this, ideally the third option is more correct. Okay, so it basically multiplex PCR will have numerous primer sets within a single PCR mixture, and this means that the products produced are of varying sizes and are each specific to different DNA sequences. Okay, and multiplexing reactions can actually be divided into two categories: single template and multiple template reactions. Both use several primers, and annealing temperatures for each of the primers must be optimized to work in a single reaction. Therefore, the design of specific primer sets is essential for the success of the multiplex reaction. The primers are usually about 18 to 22 base pairs long and have a melting temperature of 55 to 60 degrees Celsius unless the sequences have high GC content. So in this case, the melting temperature is higher. Specificity and dimerization should also be considered when designing the primers. Another requirement is enough variation of base pair length so that distinct bands are formed during gel electrophoresis. Okay, So the benefits can be substantial in labs where it's repeated analysis of you know similar types of samples and the same targets is required. So advantages include gaining more information with limited starting materials, is cost effective nature, time saving, higher throughput, and essentially by targeting multiple sequences at once, additional information can be gained from a single test run that may otherwise have needed additional reagents and more time to perform. Okay. Also, as I said, false negative results are often revealed in multiplex assays, which may have not been detected in simple PCR. This is because each product provides an internal control for the other amplified fragments. The disadvantages will be can be self inhibition among different sets of primers can occur. So primer dimers are a potential byproduct of PCR, which consists of two primer molecules that have hybridized to each other because they share a string of complementary bases. So DNA polymerase then amplifies the primer dimer competing for the PCR reagents and potentially inhibits the amplification of the target DNA sequence. So therefore, primer dimers cause a second disadvantage, which is low amplification efficiency. So generally, this makes multiplex PCRs challenging to design and means that the technique cannot be scaled easily to very large targets. Okay, so splitting up samples is often required to reduce the challenges faced by very large sample sizes. However, this in turn decreases the test sensitivity to rare variants. So using droplets or linking primers to increase specificity are alternative solutions that have been used to overcome some of these obstacles and reduce primer dimer formation. Another complication is that only a single set of amplification conditions can be set for each PCR product. Therefore, primer design for some multiplex targets can be difficult. There is a risk of favoring certain products over others, creating biases. An option is to consider several factors during primer design, such as target nucleic acid sequence, length, and GC or guanine cytosine content. So this makes it more likely that the set of amplification conditions will be optimal for all of the products that are targeted by the multiplex PCR. So most researchers now use in silico design tools on computers to simplify these tasks. So that is it. So the answer will be third. So this was the last question. So we can conclude the session for today. If you have any questions, you can ask me. And if at any point my video, I actually turned off my video so that there could be better signal. Maybe there's some network issue from my part. So if you need to refer to any part of the session today, it will be uploaded. So you don't need to worry about that. So it will be uploaded. If you have any questions, you can type or you can ask me.